Thank you, everyone, for joining us in this presentation. So today I will be presenting on our team's approach for predicting drug interactions. This is just a disclaimer. Again, this work is representative of our team's work and not of our employer. This first slide is the details of the challenge, which Bev has already gone through, so we don't need to go through this again. And then, again, in terms of our team, so my name is Thuy. I will be presenting to you guys an introduction to the challenge and our team's approach to that challenge. Ola will then go into the mechanics and the programming of this challenge, and then finally Jeff will wrap things up, and he will be providing a conclusion as well as possible steps in moving forward. Our goal was, with this challenge, was to initially determine indicators of potential drug interactions. From those indicators, we wanted to develop a model with high accuracy and value. And then from that model, we were hoping to have an output of some sort of interactive tool. So we began with several databases, and the one that we ultimately used was the DrugBank. When we looked at the data that we had, a high-level observation that we had was that with the known interactions, we noticed a higher utilization of resources for those 65 years or older who filled at least one prescription. So we wanted to develop a model to identify potential interactions in a cohort of these patients, and we ended up choosing the a cohort of patients that had major depression. And we wanted to see if the same observation from above exists in this cohort of patients. To accomplish this, we were going to perform testing of known interactions against claims data, and for this we used the IMS data. And then we were going from that data, we were using machine learning techniques to develop various models of known drug interactions. From this model, we wanted to identify a single model based on best performance. And then based on that model, we were going to see if we can predict new potential drug interactions. Again, in terms of the data sources that we looked at the Open FDA database, DrugBank, and two sites. And like I said, we ultimately went with DrugBank. And then for non-public sources, we used the IBM Market Scan database. And this, I apologize, I think earlier I said IMS, but we used the IBM Market Scan, where we looked at the cohort of patients of those with major depression who were 65 years or older. And the numbers that we use in terms of the database was from October 2015 to mid-2019. We also looked at the IMS.Givia database, sorry. And with that, I will pass this on to Ola.
Thank you very much, Thuy, for the introduction. Now, what we did next was to compare all the public data sources, the three main public data sources we used: Open FDA, two sites, and DrugBank. The idea of doing this is to see a measure of agreement between the three of them to decide if we're going to use one or two, the three of them, or the best to use in doing our analysis further. So having compared that, what we realized is there's a lot of agreement just using samples of those information we extracted at the earlier stage of the project. Comparing OpenFDA, DrugBank, and two sites, we realize there's a lot of agreement or concordance between OpenFDA and DrugBank from the sensitivity analysis you can see on the right-hand side. So based on that, we said we're going to go with DrugBank because when we extracted the data from DrugBank via API calls, the information we extracted, it came in pairs. It was going to be easy for us to use that information from data wrangling perspective to continue with any other analysis or modeling we want to do. That was why we decided to go with DrugBank as the main database here. And with the fact that DrugBank came in pairs, we decided to look at the relationship between those pairs of drugs from a network graph point of view. We looked at this basically from a dyadic point of view. A dyadic interaction mainly can exist between two objects, which can be mutual, asymmetric, or not. But in this study, we're going to focus on the mutual and the asymmetric interactions in our model from DrugBank database. If you look at the network graph right on this page, one thing, again, you realize this is just a sample of all the drugs. We did not look at all the drugs altogether because of the volume of the data. one thing you realize is based on the nodes, the edges on this particular graph, we used RS concept unique identifier label from CMS website, Center for Medicare and Medicaid Services. Concept unique identifiers are basically conditions that those drugs were intended to target. So these are just samples of what you see here. One thing you realize again is that we have a lot of arrows pointing outward here in this particular graph, indicating the fact that we have a lot more drugs out there that, could possibly interact with this sample we have on this particular view. And having done that, we decide to incorporate our claims data. We use the claims data, just a sample, 21,000 patients, people who fill between 2 to 6 drugs within the period we're looking at. That is between October 2015 and June 2019. So those people, they use these 2 to 6 drugs concomitantly within those periods. And so we decided to just give a summary, demographic summary. One thing you realize here is the fact that the female to male ratio, there's a lot of females here within the population we're looking at, 65 and above, which is not unexpected because there's a lot of research out there on life expectancy for people who are 65 and above. We designated every individual that filled 2 to 6 drugs. They were indicated in DrugBank as yes interaction, and then those that fill similar drugs between 2 to 6 compounds, but those drugs they fill, they are not in DrugBank. We designated them as no. So our case will be yes, and our control will be no in this case. This is just a high-level summary from a demographic perspective based on the claims data when we looked at those drugs from claims point of view. Again, furthermore, looking at claims data, we decided to say, since morbidity is our focus here, we want to explore all the morbidity measures essentially. We used inpatient ER visit as well as utilization per 100K as a proxy for our morbidity measure here within the claims data. And what thing we realized is that there seems to be a gap between our case and control. So back to that same 21,000 sample of patients we took from our claims data, we looked at the inpatient visit and just using the median based on the normality test we did, we decided to go with median. So if you look at the inpatient visit, it's slightly higher for those that fill or consume those drugs we extracted from DrugBank, designated as having interaction, slightly higher than those that did not consume those drugs, but they took similar drugs between 2 to 6 compounds again. Not a whole lot of difference when you look at emergency room visit. If you look at the utilization, the box plot in the middle and the center, utilization per 100K, there's a slight higher utilization for those in the yes indicator versus those in the no. We also decided to look at some other underlying conditions. By underlying condition here, we look at patients with cancer. If you look at those patients with cancer, for those that had cancer within that period, the major depression cohort, and they feed or consume any pair of those drugs we extracted from DrugBank, those people reported more adverse events within the period than those that had cancer but did not feel or consume any pair of those drugs. However, this second group, they took similar number of compounds. That shows that there's something going on from interaction point of view here. And if you look at those with chronic condition, our definition of chronic condition here are people with chronic obstructive pulmonary disease, coronary artery disease, CAD, diabetes, asthma, and congestive heart failure. So those with chronic conditions that actually feel or consume any pair of those drugs we extracted from DrugBank reported more adverse events than those that had chronic conditions but did not, even though they consume similar number of compounds. So what this is telling us essentially is the fact that directionally our claims data seems to confirm all those drugs that were designated as having interaction in DrugBank. And that whatever we're going to do with DrugBank is not counterintuitive. Based on that, we decided to say we're going to go now into our modeling. Now, having looked at the modeling from different perspectives, our main hypothesis here is that drugs with similar chemical properties behave in similar ways. To go ahead and test this out, we used NDC reference database, which contained about 73 variables and altogether almost 188,000 entries. But the final model we came up with after doing every map reduction includes our target variable of interaction, which is yes or no. So we have individual generic name drugs, and then we tag them with interaction, yes or no. And then for the explanatory variables, we decide to decompose our ATC code. ATC here is the anatomical therapeutic chemical classification coding system. So ATC level 1 represents the anatomical system, ATC level 2 represents the drug class, ATC level 3 the pharmacological subgroup, ATC level 4 is the chemical therapeutic or pharmacological subgroup, and as well as the dosage form. Now we reduce the final independent variables from 73 all the way to 5 just to conform with the law or principle of parsimony. In other words, we're trying to ensure that the final model will be economical in terms of the number of independent variables we are feeding into that model. We also perform variable of importance to see which of those independent variables is carrying the most weight. And we realize that our ATC controls about 54% in terms of weight contribution to the overall model. Within this 54%, ATC level 1, which is the anatomical system, the drugs were intended to target to the bulk of the percentage in terms of variable of importance. Now, we also decided to split our data in the test and training into 50-50. Why 50-50? We were looking at the modeling from likening to more like a forward propagation and a backward propagation perspective. In other words, we used the training to develop a model and then the test to do the prediction. And then in the backward propagation, we use the test in developing the model and then using the training to test it so that we can have a holistic, a comprehensive net new prediction of drugs. We use, of course, 10-fold cross-validation, applied 8 different kind of models, which you can see in the table on the right-hand side. The first 5, number 1 to 5, are basically assemble models. And then altogether, when we perform run the models, we came up with 6 different measures to judge the performance of our model. Those 6 measures are basically cross-validation accuracy, cross-validation kappa, area under the curve, the error rate, sensitivity, and specificity. When we compare these 8 models, and of course, as long as the assumptions of those models were fulfilled, we apply them. Our champion model for this particular exercise happened to be random forest. So we decided to use random forest in predicting net new drugs. Now, at this stage, what we've done is to predict at the individual generic name level drugs which particular drug is going to interact or not. We are yet to determine which drug they interact with. So in order to do that, what we ended up doing is to go ahead and employ similarity measure. Now, for the similarity measures, we exploit so many measures here, Euclidean, which we do within the space of metric space topology. We're talking of your Minkowski, your Mahanti, or Mahalanobis distance. We tried Tanimoto and Jacquard and Pearson. Some of these will not really work well here, even Spectra too. So we decided to go with cosine similarity measure. Why? When we applied cosine similarity measure compared to others on the drugs we extracted, samples of drugs we extracted from DrugBank, we found out that for the most part, most of them had a score that is over 50%. So based on that, what you see on your left-hand side for known interaction is just an example of one of the drugs. If you look at abacavir and tocilizumab, abacavir is used for treatment of human HIV, while tocilizumab is used for lymphoma. The model we use, they predicted both of them to have interaction, and then the cosine similarity measure between the two of them is approximately 0.55. When you look at the right-hand side of this table, these are completely net new predicted interactions, which you will not find in DrugBank, essentially. So when we predicted this, for the sulfaprothrin and then the triclomethane, the first one is used for tract infection and the other one for cancer therapy. They were independently predicted to have interaction, and then their cosine similarity measure is almost 90%. Based on that, the interactive tool we developed, which we are going to show you at the end of the presentation, we decided to choose a completely new set of case and control based on this net new predicted set of drugs that are model predicted. In other words, we want to test out what we predicted to see if we're going to observe exactly the same thing we saw at the beginning. I'm going to go ahead and hand it over to Jeff Allen to bring this home for us before we do the interactive tool demonstration.
Excellent. Hello, everyone. This is Jeff. Good day. At this point in the work, we've developed a model for prediction. We've isolated and used general utilization statistics. And we focused on several attributes of known interaction pairs dredged from publicly available resources, and then used those attributes to come up with predicted interactive pairs of compounds. The question, though, that still remains after all the dust has settled on this is, sure, we've predicted drug pairs, but can we prove something, just a little something, about the depression patients who have overlapping use of these predicted pairs, and whether or not we can discern a difference in their treatment, their utilization, general health, or sickness, compared to, say, depression patients who have no record of using the predicted pairs in an overlapping manner. And we were able to do this in the earlier part of the analysis for the known interaction pairs. That gave us confidence that true interaction may very well be evidenced in patient-level claims data. It gave us enough evidence to move on with our modeling, give us hope. Can we see similar outcomes or difference between predicted interaction compound users and those who did not use the interaction compounds? That's what we're going to explore in brief on this particular slide. So a test was in order, obviously. I took pairs that both Ola and Thuy in tandem worked together and predicted, and I searched for users with overlapping usage in the IDM market scan database. We matched our patients with interactive predictive compounds, which we will refer to as our cases, to a control or comparator group of depression patients on the basis of age and gender. For those patients who are in our control or comparator group, obviously they did not have an interactive compound overlap date, so we assigned them a random index date. And then we ensured both patient groups had adequate and analogous follow-up time. And we arrived at 14,329 patients to examine in each of the groups, the cases and controls. And you can see in the upper left-hand table there a very quick attrition report of what happens as we introduce the few criterion points that I just elucidated on. So, after doing this, we examined their utilization in a 60-day period following their index dates. And we compared utilization on the basis of encounter type from the encounter table of the Sentinel Common Data Model. The Sentinel Common Data Model is an FDA product, and what we have access to internally are several real-world databases where that we've already converted to Sentinel. The observation that we make, as you can see, cases represented in the reddish maroonish color, controls represented in the bluish color, is that in that 60-day period, these 29,000 or so patients, 14,000 roughly in each group, we see that the cases far outstrip the controls in volume of events in all venue types. And one of the next questions that we had to consider was that is there something going on prior to entering this 60-day period that we've examined here in terms of utilization that makes the cases and controls different? We already know that they've been matched on age and gender. We already know that they have major depression. So, but is there something else perhaps that we can quickly test to find out if there's something else going on in a general aggregate way to assess to see if there's any difference in their prior history? We used the Charlson Comorbidity score and we apply it to their entire database history, the era preceding their index date. Now, this history, of course, is limited to the amount of time we have available in the IBM MarketScan database in concert with the available follow-up time for each of the patients, which obviously can be different for each patient. But nonetheless, it is a condition that applies to both cases and controls. Now, if you're not familiar with it, the Charlson is a continuous measure of a patient's level of sickness or comorbidity. Occurrence of diagnoses like, for example, myocardial infarction, HIV, or metastatic carcinoma are assigned values ranging from 1 to 6 based on their severity. Those values are summed and then the patient has a score. The result indicates here, if you look in the lower right-hand table, that there is a significant, though very tight difference between the cases and controls. But it is significant as resulted by the t-test that you see right there. We see that the cases have roughly an average Charlson mean of 3.44, and the controls are at 3.06. You can also see in the bands of confidence that surround those two parameters that they're very, very stable. They're very, very tight. And, of course, more will be done on this to see if there are some strange, odd things that are preventing these from being a little more variable. But ultimately, what we see is that, and the thing to take away from this is that patients come into this 60-day evaluative period looking roughly the same in terms of comorbidities. And yet the cases far outstrip the controls in terms of their consumption of resources. Obviously, these numbers are not hard and fast conclusions. These are observations that will lead us to pursue this model. Now, in concluding, a few points to take away. This model that we've developed, particularly the part that Ola and Thuy have created that performed the prediction using machine learning techniques, is scalable to any drug database available. And what is also a nice feature about it is that it can absorb any patient-level real-world data in Sentinel Common Data Model format. We, as I've mentioned before, have several different databases that we have transformed into Sentinel. And so we can get a variety of angles from a variety of data sources. The second key point here really to take away is the importance of drug class. Particularly ATC, it was to us arriving at the results and the predicted pairs that we have. The ATC, when broken down into individual components, allows us to look at a drug's targeted anatomical system. It allows us to look at drug class, a pharmacological subgroup under the class, as well as a chemical level pharmacological subgroup that may exist below that. And finally, I just want to say that this is a prototype. We are not quite finished with this. but it has given us sufficient evidence to go forward. There is more to do. Obviously, there's a few things that I will point out here that we would like to do, and that would be a more deep clinical examination of the claims records that we've seen. We would like to further delve into whether or not the adverse events that we that have, to further define them and see if we can reconcile them with the data. And we would also like to vet our list of predicted drug pairs in such a way that it makes sense in a pharmacological, pharmacokinetic, pharmacodynamic way, pointing out things, are they too obvious or not interesting based on the presence or of other patient-level circumstances. We also want to go a little bit further beyond the dyadic. We want to explore combinations that may exist in a triadic or polyadic context. Finally, we really, really want to add more chemical attributes to our predictive model, things that get down at the molecular and bioprocess level. It could be any number of different things: atomic weight, molecular weight, monoisotopic mass, formal charge, and the Lipinski statistic. Those are things that we would like to add to make really dive down into the greater granularity, and we do expect to be continuing working on that. And that is our presentation. I'm going to turn it back to Ola for a demo. Thank you.